Hello everyone. Today we'll be performing citrate utilization test. So citrate utilization test is carried out to check whether a bacterial culture or a bacterial cell or a bacterial colony is capable of utilizing citrate as a source of carbon. So there are certain bacterial cells which are capable of utilizing citrate as the sole source of carbon. So we use citrate, Simon citrate agar over here, wherein we have citrate or citric acid as the sole source of carbon. So only those bacteria which are capable of utilizing citrate as the sole source of carbon will be growing here. And when such bacteria grows on Simon citrate agar, it metabolizes the citric acid, and through intermediates it leads to production of pyruvic acid and carbon dioxide. So the carbon dioxide produced thereby reacts with sodium and water, and that produces sodium carbonate. Now in this medium we use a particular indicator called as bromothymol blue. Bromothymol blue in acidic condition will be green in color, as you can see in this medium. Here we have citric acid, so the medium is acidic as of now, and that is why the bromothymol blue indicator that has been used over here is green in color. But when the pH of this medium becomes alkaline, then the color of bromothymol blue changes to blue, and that is actually indicative of the fact that citrate has been utilized. So what is the principle? Citrate can be utilized by certain bacteria as the sole source of carbon. Once citrate is metabolized or broken down through intermediates of oxaloacetic acid, it produces pyruvic acid and carbon dioxide. The carbon dioxide produced thereby reacts with sodium and water from the medium, and it produces sodium carbonate. Now sodium carbonate is an alkaline substance, so the pH of the medium changes to alkaline, and then the medium color of the medium becomes blue. So changes of the color of the medium from green to blue is indicative of positive result, and if the color remains green like this, then that is indicative of a negative result. Now let us perform the test. And one more thing that is essential for us to know is we prepare a slant here. Now when we prepare a slant, we must take care of the fact that there has to be a bud portion. If you see, it is completely filled in the tube, and there is a slant position wherein we have a slanted solidified medium. So we need a slant for carrying out Simon citrate utilization test. So first of all, we will heat the loop. And we will take, this is a mixed culture, we will be taking the yellow color pigmented colony that you can see, which is a distinct colony. So we will take a loop full of the pigmented colony. And then we will take the tube, we will heat it near the burner, and first of all we need to stab it. So we need to put it inside. If you can see, it goes inside, and then we will have to make a continuous stream. And then close the tube, and we will have to now incubate it at 37 degrees centigrade for 24 hours. Now we have one tube, and this is something that we had performed earlier, and if you can see, the color of this tube has changed from green to blue. So that indicates indicative of a positive result. So once the bacteria is capable of utilizing citrate, it will produce sodium carbonate, making the pH alkaline, and thereby bromothymol blue will become blue in color. So this is a positive result, and if it remains like this after incubation, then it is a negative result. That is how we interpret.